Today I am going to start molecular basis of heredity for class 12 student. What is central dogma of the molecular biology? Central dogma of molecular biology was discovered by Crick. Central dogma of the molecular biology was discovered by Crick 1958, and according to him, DNA forms RNA, RNA forms protein, DNA forms RNA. This process is called transcription, and next step is called translation. So, in last class, I have discussed transcription. Now, I would like to start translation. Translation is the second part of the central dogma. So, central dogma consists of two parts, transcription as well as translation. But in few viruses like AIDS, transcription, reverse transcription takes place. So, it was discovered by Temin and Baltimore 1970, and it is also called Temenism. What is translation? And before going to start translation, it is very important to know about the few concepts like inside the nucleus in eukaryotes DNA is found, but ribosomes are found in the cytoplasm. So, that is why they are located separately, because due to manufacturing centers are the ribosomes, and DNA gives the instruction, and DNA are permanent master document from which master copies are prepared in the form of messenger RNA. DNA forms messenger RNA in the form, DNA is the master copy which gives information in the mRNA. mRNA formation takes place by the process is called transcription, and the bases triplet sequence of the messenger RNA is converted into a specific base sequence of amino acid, and this process is called translation. Translation is a mechanism which takes place inside the cell in the cytoplasm, and you know protein factory, ribosome is called protein factory. What are the requirements? What are the requirements? Amino acid is the most important requirements. There are about 20 amino acids. Amino acids provides the raw materials, and there are various factors for initiation and other enzymes, and ribosomes are the most important requirement, and which is called protein factory of the cell. There are various types of ribosomes. Basically, there are two types of ribosomes, 70S and 80S. 70S ribosomes are called prokaryotic ribosomes. They are found in prokaryotic organisms like blue-green algae, bacteria, mycoplasma, also found in some cell organelles like DNA in chloroplast and mitochondria. And 70S is further divided, further consists of two subunit, large subunit and small subunit. Large subunit consists of 30S and 50S. They are not the additive value, they are just two different parts, and similarly 80S consists of two parts, 40S and 60S. Ribosomes are functional in state when they are associated together. If they are dissociated, they are non-functional. So, next and last important requirement is mRNA. mRNA is a thread-like structure, single-stranded, and which contains nitrogen bases in triplet form. They are called codon. They are called codon. There are about 64 codons, and out of 64 codons, one codon acts as initiation codons like AUZ. AUZ is called initiation codon. Before the initiation codon, there are some space called UTR, and at the 5 dash prime or 5 prime, and there are some termination codon like UAA, UAG, UGA, UGA. They are called termination codon. Any one of them comes and synthesis of polypeptide chain is stopped. And after the initiation codon, also there are space in the mRNA at 3 prime, so they are called UTR. UTR means untranslated region. They are located before initiation codon or and termination at 3 dash and after termination codon. So they are important requirements. And after the requirements, I will discuss mechanism of translation. Mechanism of translation is very complex and it takes place in the cytoplasm in both prokaryotes as well as in eukaryotes. The mechanism of translation can be discussed under following major head. Number 1, activation of amino acid. Amino acid in the protein synthesis provides as raw materials. They are not in active form, they are in inactive state. So, activation is required for the starting of translation or protein synthesis. Activation is done in presence of when amino acid is joined with ATP in presence of enzyme and magnesium to form amino acyl AMP complex, and pyrophosphate is released. So these energies energizes the amino acid. So this process is called activation of amino acid. Amino acid activation is very important starting point. Or mRNA, next point is the charging of tRNA, and you know tRNA contains anti-codon. Codon opposite to codon called anti-codon. And amino acid binds with tRNA, and amino acid AMP is released. So this is amino acid, and this type of amino acid is joined with tRNA, and there are various types of tRNA. So they are catalyzed by an enzyme called amino acyl AMP tRNA synthetase enzyme, and amino acid is joined with tRNA, AMP is released and enzyme is released. Third point is the activation of ribosome. Ribosome is very important and it contains 3 sites: E site, P site and A site. E site means exit site, P site means peptidyl site, A means entry. And another smaller unit, that is if they are prokaryotes, there are 30S, if they are, and large unit is called 50S, and if in eukaryotes there are 40S and 60S. So they are not the additive value. In prokaryotes 70S type ribosomes are found, but in eukaryotes 80S ribosomes are found. And activation means though both ribosomal subunits are associated together. So first of all, ribosomal subunit binds with mRNA. Five prime, 3 prime, mRNA codon is situated on the messenger RNA, and specific codons initiation chain. Initiation chain is brought about by various factors. In prokaryotes, there are 3 initiation factors, IF1, 2, 3. But in eukaryotes, there are 9 initiation factors. In eukaryotes, there are 9 initiation factors. 1, 2, 3, 4, and in 4, A, B, C, D. So this is called initiation codon. Suppose if they are joined with smaller subunit 30S and associated with 50S, so they are called active functional ribosomes. And now functional ribosomes, they are joined with 30S and 50S in prokaryotes, but in eukaryotes they are 40S and 60S. This is initiated by in presence of magnesium ions. And now activated tRNA joined with, suppose if the codon is AUZ and it will recognize methionine. It is responsible to recognize an amino acid called methionine. So activated amino acid comes and they are joined by a hydrogen bond, H bond. And methionine is recognized and carried by tRNA. So this is called activated tRNA. Now this tRNA enters inside the A site or entry site, and later on peptide, another amino acid comes, so where peptide chain is formed at P site. So another amino acid, mRNA, reprise, and now ribosome moves to the another site. Next amino acid is joined with the help of peptide bond. Peptide bond. Next amino acid is joined and ribosome moves to the next codon, and peptide bond is formed at the P site of the ribosome, and peptide bond is formed CR1H amino group and carboxylate group. One amino acid is characterized by 2 functional groups. Suppose if first amino acid and joined with another amino acid, so peptide bond is formed. Next amino acid so one molecule of H2O is released and bond is formed. So this is called peptide bond. This is called peptide bond. See, first amino acid R1, NH2, H, another amino acid comes, so this is called R2, H, COH, and so on. So that is why they are repeated. And third point, inside the chain elongation is translocation of tRNA. So in this way tRNA is released from the E site, and peptide formation, peptidal transferase, in presence of a enzyme, peptide bond is formed in between the two amino acid, and next amino acid is joined and another peptide bond is formed. So that is why they are called polypeptide chain. And ribosome moves from first codon to third codon, fourth codon, fifth codon. In this way, ribosome moves from first codon to fifth codon, and protein, a polypeptide chain is formed. If 5 prime, 3 prime, there are UTR region before initiation codon and UTR region after initiation codon. When UAA, suppose if the termination codon is UAA, so here tRNA is joined and enters at A site, and four different amino acid, 1, amino acid 2, amino acid 3, amino acid 4. In this way, they are linked together by peptide bond. And a short chain of polypeptide amino acid is called polypeptide chain. So last point is the sixth point is the termination of polypeptide chain. It is brought about by UA, UAG and UGA. Any one of them comes, and termination of polypeptide chain is brought about by termination codon. They are called termination codon. There are three termination codon. Termination codon. As a result of termination codon, tRNA is released from the E site. They enter from the A site, and polypeptide chain is produced at P site and released from E site. So termination codon responsible for the termination of polypeptide chain. As a result, a new polypeptide chain or primary chain of polypeptide is formed. Later on, they are modified. So the last point is the modification of released polypeptide chain. Released polypeptide chain is modified further and to form secondary, tertiary, and further refolded, folded and refolded with the help of folded and refolded with the help of a special protein called chaperone. Chaperone helps in folding and refolding of the new polypeptide chain. In this way, protein is produced from the primary transcript chain to. Thank you.